Okay, so this is lesson number 5, the origin of cells, and we're going to be talking about subtopic 1.5, the origin of cells. This is a pretty short subtopic as far as the to-do list from the IB goes. There's a lot of ideas and things to practice these skills, and we're going to work on that today. So our things to cover are cells can only be transformed by division of pre-existing cells. The first cells must have arisen from non-living material. The origin of eukaryotic cells can be explained by the Endosymbiotic Theory, and the application is evidence from past experiments that spontaneous generations of cells or organisms do not now occur on Earth. So here are our targets. I can analyze evidence for the evolution of eukaryotic cells from early prokaryotic cells, and I can use prefix and suffix to break down large terms such as endosymbiosis. So first we're going to look at cell theory. So do remember the 3 principles of cell theory. Take a minute, pause, and try to remember them or write them down. Check your notes, remind yourself of them. These should be something you memorize and have at the tip of your tongue to pull out to talk about biology. So the first one, all organisms are composed of one or more cells. Then cells are the smallest units of life, and all cells come from pre-existing cells. So we've talked about this before, and this is a very well-supported theory, but to become a theory in science, it has to be very well supported. But there are several different issues with it. Can you remember what the issues are with this theory? These 3 images here are meant to help remind you of some of the examples we talked about as possible exceptions to this theory. So again, pause, think about it, talk to your desk partner. So some of the issues we're talking about the size of these examples, so both with the ascending hyphae and the fungi and the large algae, the Acetobacteria, both of these have very, very large cell size compared to the norm, which makes it seem like is it possible that the cells are the smallest unit of life. And the second one that's a bit tricky are striated muscle cells because they have multiple nuclei, which again seems like every cell has one nucleus, so this is an exception. But technically, none of these examples actually break any of the 3 principles of cell theory, so we're not actually causing any issues here. So we're going to talk about some actual issues today in this subtopic. So the first one is origins. So the origin of cells is really important for us to understand and consider in terms of both evolutionary history of life on the Earth and just how these things came to be. So cell theory, one of the 3 principles of cell theory is problematized by the first cells, the source of the very first cell. So which of those principles are we questioning here? So we're talking about the idea that all cells come from pre-existing cells. If that's true, then where did the very, very first cells come from? How could the first ones ever come to exist if there was already cells there? Right, so this is the big problem with cell theory as it is. So we haven't found evidence for spontaneous generation, which is the idea that life can come from non-existing, from non-living things. We haven't found any evidence that it's actually happening on Earth, but the first cells had to come from somewhere. And if there was no life before them, they must have come from some sort of pre-existing, from some sort of non-living things. And this is one of the examples of where science doesn't have an answer, and where a lot of people go to their faith and their religion and something bigger than humanity explaining these kind of mysteries. So Pasteur did this famous experiment in 1859. We talked about it in our first lesson about cell theory. You can see the diagram right here. So to remind yourself, I want you to take a minute and again pause the video, and either by yourself or with a desk partner, I want you to write a procedure for this experiment. Remember, you have the 3 flasks, swan-necked flasks with broth, and you can see the description through the image as well as the actual pictures themselves to help you. So take a minute, pause the video, and write out your procedure, and then I will explain after. You can double check your work. Okay, so hopefully you have at this point written your procedure, and you're just listening to me to double check what you've done. It's important that you practice on your own and not just sit here watching the video and listening and not actually doing the questions to try. So the first thing is that he had these swan neck flask design that had this long neck that would allow a blockage in the flask. So he had broth in the flask, and the broth is going to be a nutrient source for any microorganisms inside the flask. And he first boiled it to kill any pre-existing organisms. So any bacteria, anything already in there, would have been killed at really high temperatures of the boiling broth. And then as the broth is cooling, the water that's evaporating out of the tube, of the flask, will then cool down and it will condense into this narrow kind of U-bend part of the flask. This creates a seal, so no new life can enter the flask. And in that case, if the swan neck was left on the flask, the broth cooled, there's no new growth. The broth stayed, this pale yellow color stayed kind of transparent. There's no new bacteria, nothing, no life just appeared because all the life that was in that flask died. Now, if we took that flask and then cut the swan neck off, so it's an easy access point for microorganisms to get into the flask through the air, then after some time we'll end up seeing growth. We'll see growth of bacteria into the flask. So this is proof that unless you have access for life to enter the flask, there's no way for life to just spontaneously appear. There has to be a source, which supports our first principle that the all cells come from pre-existing cells idea. Now, that makes sense except for the very first cells. Where did they come from? So the Miller-Urey, they did a very famous experiment of what is called abiogenesis. So think back to your prefixes we learned about in the beginning of the course. So abiogenesis, the prefix, the letter A, means what? It means not or non, and bio is life, and genesis is to make. So abiogenesis is to make from non-living things. So it's a synthesis of living material from non-living material, which is something that we don't currently see. And it's something we're trying to find evidence for in the world to better understand the evolution of life on our planet. So this is an experiment that's set up in 1952 by Miller and Urey, and they tried to create what they thought early Earth would look like. So they used equipment in the lab to set up this kind of environment, this primitive atmosphere here that had water, methane, ammonia, and hydrogen gas in it. It had electrical spark was meant to represent lightning. It had water moving through it, and water kind of was moving up as water vapor, and moving through here, and then condensing back down again. And the idea is we had a heat source and this large amount of water in the ocean, trying to kind of recreate the idea of hot oceans, this primitive atmosphere, and mixing between the two to see what would happen, to see if there's any way that we could create life. Now notice here, there is no oxygen in the atmosphere. That's important because the early atmosphere had no oxygen. It wasn't until we had the first photosynthetic algae evolving, the first chloroplasts evolving, or organisms that could do photosynthesis producing oxygen, that we started to have oxygen in the atmosphere. And that took a long time for that to happen. We'll come back to that later on the course. Okay, after 1 week, what happened? They cooled down the sample after letting it run for a little while, and they found that there were organic compounds. So organic compounds are ones which contain carbon, hydrogen, oxygen, maybe some nitrogen, some phosphorus, and they're the compounds that then can make the proteins, carbohydrates, lipids that make up living things. So that was really interesting, the fact that no, we didn't get a living thing out of it. We didn't get cells, but we did get molecules that could become cells. So we got organic molecules from inorganic molecules in this apparatus. So the formation of simple organic compounds, that's just the very first step of abiogenesis. That's what Merlin Urey did find in their experiment. But then after that, we have to have the next following steps. Those simple compounds have to then be formed into complex polymers. So simple carbohydrates, simple like sugars becoming larger carbohydrates like starches, this kind of thing, developing to larger molecules. Some of those polymers have to what we call replicate. They have to be able to copy themselves as a way to pass on information between one cell and the next. So that would be like a primitive version of DNA, some sort of primitive inheritance molecule. Then those molecules are then becoming membrane bound, and that allows them to be like similar to a cell. They can then control their internal environment, have a separate internal environment from the external environment, and allows them to start having metabolism and moving towards all the different kind of things that cells can do. So the question is, was Pasteur wrong? Well, we know spontaneous generation doesn't happen on Earth anymore, right? The temperatures are required over 100 degrees Celsius um, are very hot, and they have a reducing atmosphere to do that, and they have to have lightning. Those things really, really exist on our planet. So right now what we have is biogenesis. So if abiogenesis is creating life from non-living things, biogenesis is creating life from living things. Back to the cell theory principles. So abiogenesis is this idea that maybe the first life came from a method like this. Um, just lightning in the right conditions, the right temperatures in the atmosphere, creating small molecules, and over time, those eventually becoming cells. But right now, all the cells that exist in the world come from pre-existing cells. Now, that's how the origins of cells are kind of problematized. The other thing to think about are viruses. So viruses are another big issue we have with cell theory. And viruses are tricky because viruses technically are not considered to be alive, right? So viruses are not alive. They're smaller than bacteria. They're smaller than other kinds of cells. Um, and they don't, the 7 functions of life, they don't perform all 7 functions of life. What they can do, they can't make new viruses. Instead of doing that, they just trick their host cell into making new viruses for them in what's called the elytic cycle. I'll show you a diagram of that next. But because they can't metabolize, they can't replicate, they don't have an inheritance, they don't have, they don't have the 7 functions like we talked about, they are not able to um, live their life. But they do some things that are like cells. They react, they're able to attack a host. So viruses always had been a kind of problem in biology of they're not technically alive, but they have some traits of life. So what are they? How they come to be? This whole thing. So look here. Here is the life cycle diagram of viruses. So if we look, we can see here we have 2 cycles. So we have a bacteriophage entering attacking bacteria. This is a virus. And it's going to enter the bacteria, attack it. And what will happen is the protein coat's called the capsid. It stays on the outside, and it just injects its DNA into the host. In this case, it's a bacterium. At this point, the host and has the viral DNA inside of it. It can go into one path or the other. It can go lytic cycle or lysogenic cycle. So there's a bunch of factors that cause it to happen. If you had a chance to take a course in virology, you definitely should. It's really, really interesting. But what can happen is it goes elliptic cycle. Then what's going to happen is the viral DNA will trick the cell, the bacterium that's attacked, to then copy make more copies of the virus. Not just the DNA of it, but also make complete viruses inside using its own proteins to do so. And then after that's been enough viruses have been made, then the cell will lyse, which means exploding, bursting out, and release all those new viruses that have been made out. This is what's happening when you're actively sick with a viral infection. Now, another thing that can happen, which is kind of scary, is the virus can then enter what's called the lysogenic cycle, where it's going to merge its DNA into the DNA of its host. So what happens is it integrates its DNA into the bacteria's chromosome, and then it's going to then reproduce and copy itself normally and add to daughter cells. So then every cell that's produced through mitosis and through RNA fission of the bacterium is going to include the DNA of the virus. So it's now making many, many hundreds and thousands even of cells that contain this DNA, from the viral DNA as well as bacterial DNA. And then if the cell, something else triggers, then a cell that contains the viral DNA can then initiate the lytic cycle and produce viruses. So this is how viruses can remain dormant for a long period of time. They incorporate their DNA into your cells, and then they'll just, a viral trigger will happen, and it'll cause them to start going into the lytic cycle, making viruses and going again. So what part of cell theory does this problematize? The big thing here is that cells come from pre-existing cells. Well, viruses aren't cells, so they don't come from pre-existing viruses. They come from their host cell copying them for them. The smallest unit of life, well, technically viruses are smaller than bacteria, but they're not considered alive. But they do exhibit life-like characteristics, which is all they've been a gray zone in biology and hard to fully classify for a long time. So that's an interesting little factoid for you about viruses. Okay, here are your key ideas. So read these over, write them down, and check for understanding. If you have a question, it's a good time to ask. You can always send me an email or ask your substitute teacher if you have questions about the ideas here. Okay, we're going to talk about endosymbiotic theory next. So, endosymbiotic theory is talking about that all of our cells, that idea that cells and everything comes from a common origin, this common ancestor idea we talked about a lot about in evolution, that also fits with cells. So we have lots of evidence about how all of life had a common start and how it's diverged from there. And endosymbiotic theory is talking about that in particular for cells and for organelles in particular. So Lynn Margulis is in the picture. She came up with this theory in 1981. She presented in 1981, and she's one of the very few female scientists of her time who did this. And her theory was postulated, her theory suggested that bacterial cells, which are prokaryotes, don't forget, were predated upon by other eukaryotic cells about 2 billion years ago. And when it preyed upon it, consumed or engulfed those prokaryotic cells, what happened is it became a symbiotic relationship. So the bacteria living inside the eukaryote wasn't consumed, wasn't digested. Instead, it began to live inside of it. So we have here a simple eukaryotic cell with a clear membrane-bound nucleus engulfing a bacterium. Now, this bacterium is aerobic, which means it can do cell respiration. Over time, that's become a relationship between which now became the mitochondrion and the cells living inside. So they both benefit from the relationship with the cell. The organelle that the cell which became the mitochondrion was not engulfed. It was not taken up. It was not digested. It just became part of the larger cell. Okay. So think back to what we know from topic 4 and knowledge of ecology. Symbiotic relationship. What is that? A symbiotic relationship is any close relationship between two species. In particular, in this particular type, both organs are benefiting. Okay, this is called mutualistic relationship. Both it's a mutually beneficial situation. So eukaryotic cells, they gain ATP production, the mitochondrion, and the bacterium, it gains protection from the environment as well as carbohydrates and carbon compounds that it can then use to make ATP. So everyone's getting a benefit from this situation. So there's a variety of evidence we have for endosymbiotic theory, and because Lynn Margulis is a female scientist, female biologist, publishing a theory in the 80s, she had to have a lot of evidence to support her theory before it was going to be taken seriously in the scientific community, unfortunately. So, but the good thing is, now we have a lot of evidence for this theory. So, first thing, mitochondria and bacteria are about the same size. Look at this top image here. You can see how the size similarity between the bacterium and the mitochondrion is very close. Okay? They both divide by binary fission, so whenever a bacterium is going to divide and make its next generation, it goes through binary fission. And when the cell divides, the mitochondrion is the organelle that divides itself. It takes care of its own replication through binary fission, just like in this image here, you can see it happening. They divide independently of their host cell. So the host cell's nucleus controls binary fission for all the organelles of the cell, except for those taken in by endosymbiosis. In that case, it is the mitochondrion in all cells. It'll divide by itself under its own control. They also produce their own ribosomes. And who remembers the difference in size between eukaryotic and prokaryotic ribosomes? This means they can make their own proteins, the large number of proteins through the enzymes that are required for cellular respiration. They have their own DNA, and their DNA is much more similar in sequence and structure than to prokaryotes than it is to eukaryotes. And finally, they have a double membrane. So this is consistent with what we call phagocytosis, which is where one cell engulfs another. Look at this image here. We can see the little red cell is the one being engulfed, and the blue cell is the membrane of the cells engulfing it. And it's taking it in. The engulfed cell now has two membranes. The red one is its original membrane, and the blue outer one is the membrane it took in from engulfing the cell around it, of the host cell. So it's a double membrane. And you can see the mitochondria, it has a double membrane here as well. Okay. That's our excellent evidence for endosymbiotic theory. Make sure you write all those down and you make note of them. Diagrams are very helpful as well for this. So there's another organelle that's been hypothesized to have been gained by endosymbiosis. Take a minute, tell your desk partner, do you know what it is? It is the chloroplast. So chloroplast, if you look here, the second part of this diagram, you can find this one on Google Images. The second one we've taken in is the chloroplast, which started out as a photosynthetic bacterium and was then engulfed and then became a chloroplast over time. So eukaryotic cells with chloroplasts, also known as plant cells, engulfed have two endosymbiotic events, whereas only the mitochondrion, like animal cells, only had one endosymbiotic event in their evolutionary history. The chloroplast was a huge evolutionary advantage. Take a minute, pause video, talk to your partner. Why? Why was it such a huge advantage to have a chloroplast? Well, hopefully you had some fun about this. Chloroplasts are allowing cells to go from being consumers to being producers. They're able to then do photosynthesis and turn light energy into chemical energy, which is a huge advantage. All you need to do is be by some sunlight and some carbon dioxide, get some water, and you're good to go for making your own energy. That's a huge deal. Let's look at some examples of this. So, Hypna is an example of a protist. So, what is a protist? Remember from topic 5 or 5.3 classification. So protists are from the kingdom Protista, and they're eukaryotic organisms. There's animal-like, plant-like, and fungus-like protists. So Hypna is normally a consumer. So look at the picture here. These are two individuals of Hypna, the genus. And the left one here, this is a heterotroph. You can see it has an oral groove to take in things it consumes the environment, similar to how Paramecium does it. And this one is an autotroph. It's the same species. How is that possible? Well, when it ingests algae, like you see this picture here, it can then become a producer. The algae inside of its body are not consumed. Instead, they enter a symbiotic relationship and start to photosynthesize inside of it, sharing their sugars with their host. Now, why is this good evidence for endosymbiosis? Well, if it happened before with bacterium becoming chloroplasts, why can't it, if it's happening again now with algae inside Hypna? So if we can see it happening, it's excellent evidence that this is a real phenomenon to look at. Okay, another example is Elysia chlorotica. So this species here is a sea slug. This is not a protist. It's an animal, and it begins life as a consumer and later becomes a producer. So what's the kingdom it belongs to? I kind of blabbed and said it already, guys. What's the kingdom? It is the animal kingdom. So this is symbiotic relationship again. This only happens when it reaches the adult stage in its life cycle. And it comes to contact with a very certain algae species called Bacteria latoria, and then it will then consume them. So this is coevolution. These two species evolved together in a close relationship over many generations. So we can see this close relationship between them because of that evolutionary history. So how is this evidence for endosymbiosis? Well, again, we have an organism consuming another, and the organism consuming doesn't die. It's just it lives inside of it, still doing photosynthesis. I'll show you a little video clip of about Elysia chlorotica. Hopefully it works okay doing it this way. Hopefully, we'll see. Let me know if it doesn't work. So that's some really good evidence about the really close connections, coevolutionary connections between two species. Okay, last concept for today, for 1.5, is the universal nature of DNA and how this is important for this idea. So the fact that genetic code and DNA and its structure and the letters used for it, all those things are universal among life on this planet, is really key and really important evidence for endosymbiosis. Do anybody have an idea why? Well, if organisms want to be able to read the message and use proteins and use DNA from different species, they have to be able to understand the language. So that's the same structure, same overall function is a huge benefit. So most species share the same codons. Those are amino acid codons, about 64 of them that make it up, and they have the same meaning in nearly all organisms. And that's huge. We'll talk about that next year when we talk about topic 7 and topic 2. So you can see here we have an amino acid. Here's a 3-letter codon, UAC. The codon is AUG for the anticodon UAC, and the amino acid methionine, or MET. Because they're very similar, this means they can all communicate, all organs can read those amino acids and those codons in the same way. So remember back in topic 5 in DNA similarities, how does that provide evidence for common ancestry? Well, if they're the same DNA, that means they could have evolved from the same individuals. The more similar sequences they have, the bigger chance there is that they have the same common ancestor, and that it just diverged some traits over random mutations over time, and then natural selection caused them to diverge to new species. Okay, again, check your key ideas from today's lesson. Read them over. Any questions, good time to ask a classmate, ask your teacher. And the last thing we're going to do to help review this concept is there's a Berkeley article on the nature of science. The nature of science is a really important idea of how science works. Part of this course, we learn about different people's experiments, like Pasteur, like Miller-Urey, and they show up oftentimes in paper 2 and paper 3 questions of the IB exam. So on school, there's a link called The Nature of Science and Endosymbiotic Theory. Read the article there, the first 6 pages only, and answer the worksheet called Endosymbiosis HSW worksheet. Okay, it's just good practice working through that kind of idea and how science builds knowledge over time. Okay, I hope this is helpful for you. 1.3 is our next lesson. The quiz is going to be scheduled for next week. I'm not sure when, but you want to get that done so you have feedback before your test. Take care, guys, and hope to see you back on campus soon. Bye.